Welcome back to this next video in which we are discussing influenza. Okay, so we're currently in the process of discussing the influenza viral replication cycle. So let me just remind you what bit of this story that we've got up to so far. So we've seen that the influenza virus will bind to the surface of the cell, and we've discussed the different ligands for the hemagglutinin molecules and the hemagglutinin esterase fusion molecules. What will then happen is it will be endocytosed through a mechanism that is not currently agreed upon. So you'll end up with an endocytic vesicle here with the influenza virus particle inside it. This will then fuse with an early endosome, so the influenza virus will then end up inside an early endosome. And what is now going to happen is the pH is going to go down in the early endosome, and this is going to cause changes to occur to the influenza virus. Now remember the big picture here. This virus needs to escape this endocytic pathway. If it stays in this lumen, it's going to be annihilated. So it is not going to stay in the lumen. At least not all of it is going to stay in the lumen. The important bit that needs to escape is the genome, the 8 or 7 pieces of RNA. They need to escape. We don't care about the other parts of the virus. The genome is the important bit that needs to get into the nucleus of the cell and then start actually making proteins. Okay, so that's the next bit that we need to turn our attention to. How is the genome going to escape? And for this I need to draw a rather more in detailed picture. So we're going to zoom in here. So I'm going to draw the membrane of the early endosome here. I'm also going to draw the virus particle much more in detail. So we'll come, where should we go? We'll go here. This is a nice little space, perfect little space in fact. So we need the purple colour that we have there for the membrane of the early endosome. So I literally am imagining that I've just zoomed in on this picture. So this line here is representing this bit here, zoomed in on. And now what I want to do is draw the virus properly. So let's put the viral envelope firstly. So I'll go for the bluey colour for the viral envelope. And I'll put this a little distance away so that we can put in the hemagglutinin or the hemagglutinin esterase fusion protein bound to the sialic acid. Okay, so there's the viral envelope, and of course underneath this we'll have the M1 proteins, and something important that I'll put in straight away is the M2 ion channel. So I'll put this in, let's have an M2 ion channel in here. And remember, importantly, the M2 ion channel, it's going to conduct protons beyond the M1 shell. So let me now put in the M1 shell. We'll do this in blue. So here are the M1 proteins just underneath the viral envelope here. And I'll complete the whole thing up. Just bear with me a moment whilst I complete this. So remember, just a little bit of revision whilst we're at it. There are 5,000 of these M1 proteins roughly making up this shell, and the purpose is to hold the viral envelope in that spherical shape. And remember, the M1 proteins are also called the matrix proteins. Okay, so just labeling a few bits up here. We've got M1 there in blue, and here is the M2 ion channel over here. And remember, all 3 of the viral species have an M2 ion channel. Now what I want to put onto the picture is the hemagglutinin molecule, or the hemagglutinin esterase fusion molecule. Now, as far as this picture is concerned, it doesn't matter whether we're talking about hemagglutinin or hemagglutinin esterase fusion, because all I'm going to show is a spike in the viral envelope bound to something that is now in the membrane of the early endosome there. So, what colour should we go for? Red, I think. So here is this protein complex here. So that is either hemagglutinin or it's the hemagglutinin esterase fusion protein complex. And it's going to be bound, of course, to the ligand molecule that will be on some glycolipid or glycoprotein that is now in the membrane of the early endosome. So just to simply show this, we'll have something here. We can imagine that that's maybe a glycoprotein that it is bound to. And of course, in reality, there will not just be one of these. And in fact, I might try and show this. Let's have another one here. So here's another glycoprotein. Here's another glycoprotein with either sialic acid or 9-O-acetyl sialic acid bound to it. And let's have multiple of the hemagglutinin and hemagglutinin esterase fusion proteins bound. So, like so. Okay, so there's our hemagglutinin or hemagglutinin esterase fusion proteins bound to the ligand that is now in the membrane of the early endosome, but which originally would have been in the plasma membrane. Okay, the final thing that I want to put on here, because it's important, is the ribonuclear proteins, and as I did previously, we'll go for violent, vivid purple to show these. So, of course, here I will show the influenza A virus or the influenza B virus. I'll put 8 of them. If we were dealing with influenza C virus, of course, I would have to put 7. But we'll put 8. 1, 2, 3, 4, 5, 6, 7, 8. And remember, these are not just the strands of RNA, they are the strands of RNA with loads of nucleoproteins, and also with the RNA-dependent RNA polymerase, which is made of those 3 proteins, PA, PB1, and PB2. Excellent. Of course, things that I haven't shown here because they're not necessary for this portion of the mechanism is neuraminidase, if we were talking about influenza A or influenza B. I'm certainly not bothering putting NB on there for influenza B, and I haven't shown the non-structural protein 2 either. Okay, these are the important bits for what's about to happen. So, we want to see how are these ribonuclear proteins here going to escape from the early endosome. Remember, this is the early endosome lumen. They need to get out here, which is where the cytoplasm is, and I might just label this up. So over here is the cytoplasm. This is where they want to be, and then they can go to the nucleus. They do not want to be in the endosome lumen because this is where eventually the lytic enzymes are going to arrive to destroy them. Okay, so how do they escape? What we know is about to happen is the pH of the endosome lumen is going to fall. Proton concentration is going to go up and up and up, and proton concentration going up and up and up causes proteins to change their conformation. Now, we already know it's going to cause the M2 ion channel to open. I'll come back to that in just a moment. Firstly, I'm going to talk about hemagglutinin, and then we'll talk about the M2 ion channel, because hemagglutinin, or indeed hemagglutinin esterase fusion, both of them, they undergo conformational changes in response to the low pH in the endosomal lumen. Now, what's going to happen is the hemagglutinin 2 or the hemagglutinin esterase fusion 2 polypeptides within the trimer are going to change conformation, and they're going to stick out something called a fusion peptide that is going to stick into the endosomal membrane. So let me just see if I've got my, I do, excellent, it copied well. Right, so here, remember, is the old picture of hemagglutinin for influenza A virus or influenza B virus. Remember, we describe it as a trimer of these dimers of hemagglutinin 1 and hemagglutinin 2. Now, the hemagglutinin 2s are the ones that are in the membrane, the viral envelope membrane, whilst the hemagglutinin 1s are stuck on top, and they're the ones where the actual binding site for the sialic acid is. What's going to happen is when the pH goes down here, which is the endosomal lumen effectively, these hemagglutinin 2 proteins are going to change conformation and they stick something up. So I'm going to try and show this. This is going to look awful, but I'm going to show it anyway just to make sure that the concept is clear. So what will now happen is, let's show it for this one at the front first. It's now going to stick a bit up, like so. This was previously bent down, you can imagine, in this structure, and now when the pH goes down, it changes conformation and a great big branch of the polypeptide sticks up. And this branch has a name. This is called the fusion peptide, and the reason it's called that is it's going to mediate the fusion of the viral envelope with the membrane of the early endosome. Now, it's not just that one that's going to stick up a fusion peptide, all 3 of them are going to do it, so I might try and show it. Here is the one over there's fusion peptide, and here is the one over there's fusion peptide. Okay, so all of the hemagglutinin 2 polypeptides stick up these fusion peptides, and where are they going to stick them into? Well, here is going to be the membrane, the phospholipid bilayer of the early endosome in our other picture. Now, it's the same for hemagglutinin esterase fusion. The hemagglutinin esterase fusion 2 is going to do exactly the same thing, so each of these 3 hemagglutinin esterase fusion 2 polypeptides is going to stick up a fusion peptide that will stick into the membrane of the early endosome, and which is going to mediate the fusion of the early endosome membrane with the viral envelope. So, just for completion, Let's draw it on here so that I'm not being unfair to hemagglutinin esterase fusion 2. Here are their fusion peptides as well. So, going back to our other picture here, each of these represents a trimer of hemagglutinin or hemagglutinin esterase fusion. So each of them is going to have 3 fusion peptides that are going to stick up, which I'll show here. In fact, I'll get a thicker pen to show the rest of them. So here they are. 1, 2, 3. 1, 2, 3. So when the pH goes down, these hemagglutinin or hemagglutinin esterase fusion proteins change conformation and fusion peptides stick into the membrane of the early endosome. And what this causes is it causes fusion of the membrane of the viral envelope with the early endosome membrane. So what's actually going to happen, to draw this separately now, and we'll go here, what's going to happen is this is the early endosome membrane here. What's going to now happen is it's going to fuse with the viral envelope membrane, like so, and this occurs because of these fusion peptides disturbing the early endosome membrane. So just to motivate this a little bit more, when you stick these in, it disturbs the membrane. The membrane is going to go into flux. It's going to start moving around a lot. And what can then happen is it can end up fusing with the membrane of the viral envelope, which is very, very close by. So what can end up happening is this becomes continuous with this, and this becomes continuous with this, just as I've shown here. So the viral envelope has joined with the early endosome membrane, and that is mediated by the hemagglutinin and hemagglutinin esterase fusion proteins. So that's viral envelope fusion. Now, there's a problem still here, because of course what we want is these ribonuclear proteins to escape into the cytoplasm. So this looks excellent. We've opened up a nice little channel through which potentially the ribonuclear proteins can escape from the inside of the virus into the cytoplasm up here. There's still a problem though. The M1 coat is all over the place, blocking these things from getting out at the moment. And this is where the M2 ion channel comes in. So, when the pH goes down, proton concentration goes up in the early endosomal lumen, that's going to cause the M2 ion channel to go into the open state, and of course it's going to conduct protons into the inside of the virus. So protons come into the inside of the virus, and this of course means that pH goes down inside the virus. Not just does it go down around the virus, but it's also going to go down inside the virus, and this means that proteins are going to change conformation in the inside of the virus. In particular, the M1 proteins are going to start changing conformation, and this causes the M1 proteins of the M1 matrix here to start falling apart. So the matrix starts breaking down, the shell starts breaking down. In addition, they stop binding to the ribonuclear proteins, and this is something that I don't think I've actually told you at any point before. The M1 proteins are capable of binding to the ribonuclear proteins of the genome. Indeed, this is how the whole thing assembles. If the M1 proteins weren't capable of binding to the ribonuclear proteins, how on earth would you end up forming a beautiful M1 shell like this around the ribonuclear proteins so perfectly like that? In order for this to happen, you do have to have the M1 proteins able to bind to the ribonuclear proteins in the center, so that you do end up with the ribonuclear proteins in the middle of the M1 shell. So, when protons come in to the inside, not only does it break apart the interactions between the neighbouring M1 proteins, and therefore the shell breaks down, but it also breaks apart the interactions between the ribonuclear proteins and the M1 proteins. So this is just perfect, basically. Now the shell is breaking down, the ribonuclear proteins are no longer bound to the M1 proteins. What can now happen? They can leave through this little fusion pore that we've got. So that's a nice little piece of terminology that I feel I should add on to this diagram. This little pore here is called the fusion pore. So here we go. So now the ribonuclear proteins can escape through this fusion pore, and then they're in the cytoplasm of the cell. And going back to our other picture over here, once they're in the cytoplasm of the cell, and I might even draw this on here, so we've just been through the mechanism by which the ribonuclear proteins will escape from the early endosome into the cytoplasm. So I'll just draw one of them here to represent all 8 or all 7 of them if we're dealing with influenza C virus. And now what's going to happen is they will go from the cytoplasm into the nucleus. And there you are. This host cell is now infected. It has the ribonuclear proteins of the influenza virus genome inside its nucleus, and the virus is very happy now because it can use the cell as it likes to now produce loads more influenza virus particles that are very, very similar to it, almost genetically identical to it. As we'll discuss, the copying, the replication of the viral genome is usually not perfect, so usually you do get slight, single nucleotide mistakes in the copying of the viral genome, so each new influenza virus that you produce will be slightly different to the previous one, but not majorly different at all. It might be that in the entire genome there's one nucleotide different. Okay, but still, they are slightly different, and this is the mechanism of the antigenic drift that we'll come on to right at the end of the video. Okay, so let's do this then. So we now have in the nucleus all of the ribonuclear proteins. So let's draw another picture of this. So we'll have our nucleus drawn here. So this can represent the nucleus of the infected cell, so I'll just label it up. This is the nucleus. And now we have our 8 segments of the genome, or 7 segments of the genome if we're talking about influenza C virus. 1, 2, 3, 4, 5, 6, 7, 8. They're all in the nucleus together. So what happens next? Well, this should be fairly obvious. What do we need to do? We need to make new virus particles. Remember our picture of the virus particle. The virus particles consist of the ribonuclear proteins and then the bits around it, the M1 shell, and then the viral envelope with those proteins in, M2, hemagglutinin, neuraminidase, etc., the membrane proteins. So in order to make new virus particles, we need to synthesize loads of new virus proteins. We also need to copy the pieces of RNA. Now, luckily, we have brought the perfect enzyme along. Each of these pieces of RNA has an RNA-dependent RNA polymerase, and this is exactly what it needs. So what's going to happen is each of the pieces of RNA are going to have the complementary piece of RNA to them made. So let me just draw this out. So let's let this red line actually represent one of the pieces of RNA. So this is the single-stranded RNA of one of the ribonuclear proteins. And remember, it's negative sense single-stranded RNA. Now, I will remind you, because it's really important that you do understand this, what the negative sense means is that it cannot be used to translate. You cannot put this for a ribosome and just expect to get some wonderful protein out. What you need to do is take the complementary piece of single-stranded RNA, which I'll draw here in green. So we make the complementary piece of RNA here, which will then be positive sense. This is the right piece of RNA to actually use for translation. This piece you can then send into the cytoplasm, and it can function as a piece of mRNA, a piece of messenger RNA. It can be translated and it will make whatever protein is coded for on that piece of single-stranded RNA. So if we were talking about genome segment 1, it would then be able to make the PB2 protein, etc. Okay. So that's the first thing that's going to happen. All of these pieces of RNA in these ribonuclear proteins are going to be copied to make the complementary piece of RNA, the positive sense piece of RNA, which is then going to be sent out of the nucleus into the cytoplasm and will actually make that protein. So we're going to start making our proteins. In addition, what we can also do with these positive sense pieces of RNA is use them to then make another piece of negative sense single-stranded RNA. So we can break this piece apart from the negative sense bit that we use to synthesize it. So here it is. And then the enzyme, the RNA-dependent RNA polymerase, can then synthesize another piece of single-stranded RNA that's complementary to that one, and therefore we have copied this original piece, and that is how we're going to replicate the viral genome using the positive sense RNA that we've produced. So the positive sense RNA has two functions. One, it's going to be used as the mRNA to actually make viral proteins, and secondly, it's going to be used to copy the viral genome, and we need to do both of those things to make new virus particles. So, let's now go over the synthesis of the new proteins, the new viral proteins. So this is the complicated bit because there are quite a few of them that we need to go through. So we'll categorize it up. So, we are going to use the mRNA here that we produce, the positive sense RNA, which is now going to be used as mRNA. It's going to leave the nucleus, of course, it's going to go into the cytoplasm where translation can occur, and we're now going to make the proteins that are coded for by our genome. So we can categorize the proteins now into different categories. There are the membrane proteins, firstly. And it's important now to categorize these into different categories because they're going to follow different paths. So, the membrane proteins. Now, of course, this differs depending on whether we're talking about influenza A virus, influenza B virus, or influenza C virus. So let's just split it up then. So, for influenza A virus, what are the membrane proteins? What are the proteins that we need to send to the membrane? And I should just say something to motivate this. When we synthesize new viral particles, the viral envelope is going to come from the host cell membrane. So, if we want hemagglutinin proteins or neuraminidase proteins or whatever to be in our viral envelope, we need to produce them inside our host cell and send them to the membrane of that host cell. So that's what I'm now talking about, these membrane proteins being sent to the cell membrane of the host cell. So, influenza A virus, what are the membrane proteins there? Well, of course, we have the hemagglutinin protein, coded for on genome segment number 4, remember. We have neuraminidase, those are the two easy ones to remember. Then we have the influenza A M2 ion channel, so I'll put that there, AM2. And then, do we have any more? Well, the M1 protein we'll classify as separate. Are there any more? I don't think there are any more for influenza A virus. Let me just go up to my picture and thusly check. Here we have our picture of influenza A virus. No, because the MB protein is one that's specifically for influenza B virus. So those are the only 3 for influenza A virus. And let's now do influenza B virus in a different colour here. Influenza B virus is exactly the same as influenza A virus, but it's got another membrane protein. So it will have hemagglutinin, neuraminidase, and these will all be being synthesized now from the mRNA inside the cell. Of course, we'll have the BM2 ion channel, and then we'll also have that silly MB ion channel as well that will be synthesized. So what will be happening with these membrane proteins is we'll be synthesizing the proteins and then we'll be sending them to the cell membrane, and in that journey they will be trimerized or tetramerized. They'll be joined together to actually make the final protein complex. So in the case of hemagglutinin, for instance, the hemagglutinin 0 polypeptide will be cleaved into hemagglutinin 1 and hemagglutinin 2. They'll join together by disulfide links, then they'll be trimerized together, and the whole thing will be sent to the plasma membrane. So overall, we're manufacturing these membrane proteins, and they're going to end up in the cell membrane of our host cell. Then to complete this up, let's put influenza C virus on here. So ICV here. So for ICV, hemagglutinin and neuraminidase have been combined. So we have the hemagglutinin esterase fusion protein, which is the membrane protein, and then we have the CM2 ion channel, and that's it for the membrane proteins. So just to draw now a little picture of this, let's get this colour here. So what will be happening, we'll draw it for influenza A virus. This is now the host cell membrane, so host cell plasma membrane, which I'll just abbreviate down to plasma membrane, or PM, sorry. So this is the extracellular side, this is the cytoplasmic side, and now once the virus has gone in and we're now synthesizing these membrane proteins, they're going to end up in the host cell plasma membrane. So drawing these on, we'll have hemagglutinin here in red, so there's that red spike. Then we'll have neuraminidase here in yellow as the mushroom shape, and of course we'll also have the M2 ion channel, which we'll do in blue. So here is the M2 ion channel. So My overall message is that the membrane proteins of the virus are going to be synthesized and they're going to be sent to the cell membrane. And specifically, they are sent to the apical cell membrane of the host cell. So they're going to end up on this side of the cell membrane, so that new viruses that are going to be budding off from the cell are going to go back into the lumen of the respiratory tract rather than go downwards into the tissue underneath. And that's important. This is the reason that influenza is not a systemic infection. If they went downwards, then they'd be going into other portions of the body potentially. They'd be potentially tunneling down towards blood vessels, and then it would be going towards a systemic infection. But they don't normally do that. Usually, they send the proteins to the apical surface, and new virus particles will bud off the apical surface and go back into the respiratory tract, go back to where they came from, and that's why it only usually causes a local infection rather than a systemic infection. Okay, so that's an important point to note there. So, we synthesize the membrane proteins that we've talked about now. We're also going to synthesize the M1 protein. We'll deal with the M1 protein next, and then we'll deal with the proteins that are going to go to the nucleus, which is the other ones. And then finally, we'll talk about the NS1 protein, the non-structural 1 protein. Okay, so let's do M1 protein, which I'll put in a separate category on its own. So again, what will happen is we will synthesize positive sense RNA from the single-stranded negative sense RNA of the genome. Some of this will go into the cytoplasm and function as mRNA, will be translated by ribosomes, so you'll actually produce the protein that it coded for. And that is when, the seventh viral genome segment is translated, you will get M1 proteins in the case of influenza A and B. In the case of influenza C, remember the M1 gene is on the sixth viral genome segment rather than the seventh. Okay, so you produce these M1 proteins in the cytoplasm of the cell. What happens to them next? They go up to the apical surface as well, and they start binding to the tails of these membrane proteins. So in the case of influenza A virus, they start binding to the tails of the hemagglutinin proteins, the neuraminidase proteins, and the M2 ion channels. So I'll show the M1 proteins then in this green colour. So they're going to start binding to the tails, like so, of these proteins. And what this results in is it results in clusters of the membrane proteins forming. So what happens is all of the viral membrane proteins end up clustering into little regions of the apical membrane, which is exactly what you want them to do, because we want to bud viruses off certain places of the apical membrane where there are loads of these viral membrane proteins there. We don't just want to get a few of them. If they were evenly distributed out across the entire apical membrane, then, the concentration of them would be far too low, and in the viral envelopes of the new viruses that we were producing, you'd end up with far too few hemagglutinin proteins and neuraminidase proteins. So we want them all to be concentrated in little patches. We don't want them to be evenly distributed, and this is what the the M1 protein does. So in pink here, I'm drawing some little patches of the membrane which can have loads of clustered membrane proteins of the virus all joined together on the cytoplasmic aspect by M1 proteins. So the M1 proteins cause these clusters of the membrane proteins to form in the apical membrane. And in these clusters, the cell's own membrane-bound proteins, they will be eliminated from these clusters. They're not allowed to stay in these clusters. So these clusters will just be of the viral membrane proteins and not the host cell membrane proteins. So this is absolutely perfect, basically, for producing new viral envelopes from these clusters. Okay, so M1 goes up to the cytoplasmic aspect of the plasma membrane, causes the clustering of these membrane-bound proteins together, so that we have clusters where loads of these proteins are really concentrated and there are no of the host cell proteins that aren't viral proteins in those areas. So that we're getting ready for budding off new viruses there, creating new viral envelopes with underlying matrix. Okay, brilliant. So let's now go to the next class of proteins. So the next class of proteins is the ones that are going to have to go to the nucleus and are involved in either forming new ribonuclear proteins directly or are involved in taking the new ribonuclear proteins out of the nucleus. Hint, non-structural protein 2, also called nuclear export protein. So let's call these proteins the nuclear proteins. So let's have some interesting colour for them. We'll have yellow now. So next class of proteins that we're going to produce are the nuclear proteins. And these are the same for all the different species of influenza virus. So it was most complicated when we were talking about the membrane proteins because those are the ones that differ for the different species of influenza virus, whereas the nuclear proteins are exactly the same for the 3 different species. So, what proteins are we going to send to the nucleus then? Of course, we're going to send the nucleoproteins that we produce, MP, coded for by viral genome segment 5. They're all going to go to the nucleus. Also, the ones that are going to make the new RNA-dependent RNA polymerases, they need to go to the nucleus. So they are PB2, PB1, and PA, coded for by viral genome segment 1, 2, and 3, respectively. So all of those go to the nucleus as well. The next one that goes to the nucleus is also the non-structural protein 2, or also called the nuclear export protein, the NEP. So these 5 are going to go to the nucleus. We'll talk about the final protein, non-structural protein 1, in a category on its own in just a moment. So, what's going to happen then? Remember, in the nucleus, we are synthesizing new copies of the genome. So all of the pieces of RNA here are being copied, and we've gone through the mechanism by which we do that. We produce a complementary strand only to produce a complementary strand to it, and therefore we end up with a copy of our original piece of RNA. Now, the RNA-dependent RNA polymerase, this trimer of PB1, PB2, and PA, it's not brilliant. It makes mistakes reasonably regularly, and therefore, when it copies the viral genome, you can expect mistakes will occur quite regularly. So there will be often, if you take your original strand and compare it to this copy here, you might find one nucleotide in the strand has been replaced with a different nucleotide. So it does make errors at a rate much higher than human equivalent enzymes would ever make. So the human DNA-dependent DNA polymerases are fantastic at not making errors, whereas these RNA-dependent RNA polymerases that the influenza viruses are using are much more prone to mistakes. But that's not necessarily a problem for influenza virus. As we'll see, it leads to antigenic drift, which is actually very helpful for the virus. Okay, so the new viral genomes that are being produced then, we'll call them genetic copies of the original ones, but we understand that there will be mistakes, and this is going to lead to gradual changes in the viruses over a long period of time. But we'll come back to that when we discuss antigenic drift and antigenic shift. Okay, so what's going to happen is the new copies of the viral genome then, they need to now be packaged up. So they will get loads of copies of nuclear protein bound to them, all the way along them, and then they will get a copy of the RNA-dependent RNA polymerase bound to them. So they will produce ribonuclear proteins. So inside the nucleus, we are producing more copies of the ribonuclear proteins of the viral genome. Now, in order to get those ribonuclear proteins now to the plasma membrane, which is where they need to go in order to produce new copies of the virus, they need to get out of the nucleus, and for this, the nuclear export protein, or the non-structural protein 2, is necessary, which is why this is sent to the nucleus. So what's going to happen is, let's say we have our new ribonuclear protein here, and remember, this rectangle is representing the full ribonuclear protein, so the RNA bound to loads of copies of the nuclear protein, twisted up in that silly way with the copy of the RNA-dependent RNA polymerase bound to it. So this is now in the nucleus, and we'll have all 8 of them, or all 7 of them if we're dealing with influenza C virus. What's now going to happen is nuclear export protein is going to bind to each one of them, so each one of them will get a nuclear export protein bound to it. So here it is in yellow, This is representing the nuclear export protein, and this will export it from the nucleus. It will take it into the cytoplasm, and then what will happen is the new RMPs will make their way to the cell surface. So they will move now from the nucleus to the cell surface, and of course you'll need a full complement of the genome, and it's not understood how this actually works so well. Initially, when we first worked out the replication cycle, we thought that what must happen is it must just be random, i.e. the nucleus will start chucking out new ribonuclear proteins of all the different types, of all the different genome segments, and then it must just be random that they will then go and bind to the M1 proteins, and then some of the viruses that bud off will end up with the right full complement of the viral genome, and most of them would then end up with the wrong complement. You know, they'd end up with not enough of the viral genome segments, only with partial only with partial some of the viral genome segments, maybe 5 of them rather than 8 of them. So therefore, most of the influenza viruses budding off wouldn't be actually useful at all. They wouldn't be able to do the job. They're useless particles. It turns out that that is not what happens. It's incredibly good. Somehow, when the new viruses bud off, they get the perfect copy of the viral genome. Each of them gets all 8 of the viral genome segments, or all 7 of the viral genome segments if we're talking about influenza C virus. We don't understand how on earth it achieves this feat, but it does somehow. So, what's going to happen then is, let's draw some arrows. So, out come the ribonuclear proteins, all 8 of them or all 7 of them, and they're going to make their way through the cytoplasm. So this is the ribonuclear proteins pathway now, going from the nucleus to the plasma membrane, the opposite of what happened here. And I'll just add on to this picture that in order to get them out of the nucleus, you need the nuclear export protein to be bound to them. So here come, let's just follow the story for making one new virus. Here come all 8 of the ribonuclear proteins, bound each of them to a nuclear export protein which mediates them getting out of the nucleus. They make their way through the cytoplasm. They then go and bind to the M1 proteins underneath the membrane in one of these clusters. So we'll come over here, and what will happen is these ribonuclear proteins will bind to the M1 proteins underneath, and then what happens, and I'll just add on to this picture, this ribonuclear protein has a non-structural protein 2 bound to it as well there, so let's just label this up. So this is non-structural protein 2, and this is the ribonuclear protein in pink here, and then what happens is the entire thing buds off, so the entire membrane bends outwards this time, evaginates. So I'll try and show this quickly. So the membrane at the moment is flat like that. What's going to happen is it's going to evaginate outwards like this, and that stalk connecting the new virus that is forming here to the plasma membrane is going to gradually break, and it will go like this, and then the whole thing will bud off and you'll have your new virus particle here. Okay, so this is a basic picture to show the budding off process. So this is called the budding process by which we produce the new viruses. So this is budding off. And of course, on the inside of the virus, what are you going to end up with? Underneath the membrane, you've got all of the M1 proteins here, and then the M1 proteins are bound to the ribonuclear proteins with the non-structural protein 2 bound to them. So the ribonuclear proteins are going to end up inside here. You'll complete up the M1 shell here, so you will end up with perfect new virus particles. You end up with a viral envelope with loads of copies of the viral envelope proteins in. Underneath that, you'll have the M1 shell, and inside that, you'll have the ribonuclear proteins along with non-structural proteins 2 bound to them. Now, what I want to do before coming on to the non-structural protein 1, I will come on to the non-structural protein 1 to finish the story, but I want to say something before I forget to say it, which is the function of the neuraminidase protein, which we've waited so long to find out about, or the esterase enzyme sites in the hemagglutinin esterase fusion protein. So, they are important in this budding off process. And where do they become important? They become important in cutting off sialic acid molecules from glycoproteins and glycolipids on the surface of the cell. So we'll start off the story for neuraminidase and then I'll come on to hemagglutinin esterase fusion. So remember, for influenza A and influenza B, the hemagglutinin molecules bind to sialic acid molecules on glycoproteins and glycolipids. When the virus is budding off and there are loads of hemagglutinin molecules in the new viral envelope, and I'll just mark these on here. So these little red spikes here, these are representing the hemagglutinin molecules in the budding off virus. There's a real risk that these hemagglutinin molecules are then going to start binding to sialic acid molecules on the surface of the cell that is already infected. That is not what the virus wants to do. It does not want to enter an already infected cell because there's no point. That cell's already infected, and indeed the cell will die very quickly once it's been infected. So it certainly doesn't want to enter a cell that is already being exploited so badly that it's on its deathbed already. So it wants to go into a fresh new cell that no one has infected yet and infect that. So what happens in order to prevent these hemagglutinin molecules binding onto sialic acid residues around this place where the virus is budding off is the neuraminidase protein cleaves off sialic acid residues from the terminal positions of the oligosaccharides of glycoproteins and glycolipids in the surrounding membrane. So remember, the hemagglutinin can bind to sialic acid residues when they're in the terminal positions of the oligosaccharide chains on glycoproteins and glycolipids. What the neuraminidase is going to do, and I'll just put them on here in, let's get that yellow colour back again. So let's have some neuraminidase. So there's a neuraminidase. Try to draw it in the mushroom shape. There's another neuraminidase, here's another neuraminidase, and here's another neuraminidase. What they're going to do is they're going to cleave off sialic acid residues from this terminal position. So they are going to cut the bond that links the sialic acid residue to the rest of the oligosaccharide, so that in this surrounding portion of membrane, there are no sialic acid residues bound in that terminal position on the glycoproteins and glycolipids, and therefore the hemagglutinin molecules will not bind to them because they're not there. Now remember, neuraminidase is a tetramer, and each of the monomers in the tetramer has its own enzymatic site. Therefore, each neuraminidase protein is capable of catalyzing 4 of these reactions at once. So they're going to cut lots of those sialic acid residues off. So this is where neuraminidase is really important in stopping the budding virus particle attaching onto the cell that it is budding off, the already infected cell. It cuts off the sialic acid residues so that the binding site has gone, and therefore the virus will bud off successfully and go off and infect a new cell. That's the function of the neuraminidase. And if these enzymes are inhibited, which indeed we have drugs that can do that, called neuraminidase inhibitors, tamiflu and relenza, proper names oseltamivir and zanamivir, work by inhibiting neuraminidase enzymes. And when you do that, the viruses end up going back into the cell that they've just infected, and it really messes up the viral life cycle. So they are very effective drugs. More effective, actually, than the M2 ion channel inhibitors, the adamantines. They're the ones that are actually used, tamiflu and relenza, rather than amantadine and rimantadine. But we will, nevertheless, at the end of the video, go over the mechanism of amantadine and rimantadine. So even though I've told you the mechanisms, I will summarise it and write it out properly later on, and we'll do the antivirals officially. Okay, right, so that's neuraminidase. Let's just now talk about hemagglutinin esterase fusion. So neuraminidase of both influenza A and influenza B does this. Now let's talk about influenza C. So remember, influenza C just has the hemagglutinin esterase fusion protein. So forget this, just have this here. And remember, that's a trimer, and the hemagglutinin esterase fusion 1 polypeptide has this esteratic site. Now, also remember that hemagglutinin esterase fusion binds to something different to sialic acid. It binds to 9-O-acetyl sialic acid. Now, you might think, oh, well, that enzymatic site in the hemagglutinin esterase fusion protein, that's just going to cut off the 9-O-acetyl sialic acid. And it would be nice and simple if that was the case, because then it would be beautifully analogous. However, they do something more complicated. They cut off the 9-O-acetyl group to make these just sialic acid, and then the hemagglutinin esterase fusion binding sites can't bind to it anymore because they don't bind to sialic acid, they bind to 9-O-acetyl sialic acid. And that is why the enzymatic sites of the hemagglutinin esterase fusion protein are called esterase sites because they are breaking an ester link. When you make a 9-O-acetyl sialic acid molecule, you remember, take the sialic acid molecule, so this thing here with an acetyl group attached to the amino group, and then you put another acetyl group onto this this alcohol group coming off the 9th carbon. And that bond is an ester link. When you attach a carboxylic acid group to an alcohol group, that's an ester link. When you break an ester link, you are called an esterase. So that is why hemagglutinin esterase fusion is called hemagglutinin esterase fusion, because the enzyme activity is esterase function, whereas the neuraminidase enzyme, also called sialidase enzyme, is breaking the whole sialic acid molecule off the oligosaccharide. It's breaking glycosidic links there, and those are not ester links, so you cannot describe neuraminidase or sialidase as an esterase. So they are catalyzing different reactions. Let me just summarize that because it is important to understand. So, influenza A and influenza B have the neuraminidase enzyme. Influenza C has the hemagglutinin esterase fusion, the esterase sites of that protein. And these are really important for virus budding off from the infected cell. They're really important to make sure that the budding viruses do not re-enter the infected cell that they are coming out of. So, neuraminidase enzymes, they will cut sialic acid residues off glycoproteins and glycolipids in the surrounding portion of membrane where the virus is budding off, so that it doesn't just then attach straight back on to the plasma membrane it's come off and then re-enter that cell, so that it goes off to another uninfected cell. Hemagglutinin esterase fusion, it cuts off the acetyl group off the alcohol group of the 9th carbon of the sialic acid molecule, turning it into sialic acid, which the hemagglutinin esterase fusion binding sites cannot bind to, and therefore the virus cannot bind anymore to this, and therefore again will go off to find a new, fresh, uninfected cell. And that is the viral life cycle. We have now seen how we produce new viruses. One virus entered, and now loads of new viruses will be coming out, because we will be producing loads of new copies of the genome. So loads of RNPs will be coming out. We'll be producing loads of copies of the protein to produce loads of these clusters here, and therefore we'll be getting loads of these new influenza viruses coming off the apical surface, and they are genetically identical to the original one, although we know that that isn't quite true. There will be little copying mistakes, so actually all the new influenza viruses have tiny, minuscule differences that might not make any difference at all, or might make a gigantic difference depending on the nature of the actual single nucleotide mistake, where it is, if it's in a gene, if it causes a massive mutation in that gene, who knows. But all of the new influenza viruses will be slightly different to the original one, genetically definitely, and potentially in phenotype as well, i.e. in the actual proteins that they can code for with their new genome. Okay, the next thing to say before we go on to the non-structural protein one, I will come back to the non-structural protein one, is that this is not good for these epithelial cells whatsoever. Having a virus come in and exploit all of their machinery to make thousands and thousands of virus particles that are coming off the apical surface of the cell, it's not good for the cell at all. It kills the cell. The cell will die because of this happening. It can't survive this sort of infection. And typically, after the cell has produced thousands of new fresh virus particles, that's when it will start to die. So it takes a while before the cell has been knackered enough to actually die. So after producing maybe thousands of the new virus particles, the cell will die. So I'll just put this here. This cell is doomed to die. It cannot survive this process of being exploited like this. But before it dies, it will have produced thousands of new virus particles. So one virus particle potentially went in, thousands are coming out, and they're coming out onto the nasal epithelium. They are now going to be able to spread to other nasal epithelial cells, and then they're going to be able to spread down the respiratory tract further to the next bit of respiratory epithelium, which is the nasopharynx, and then down further, but we'll discuss that all more later. Okay, let's now discuss the final protein that is coded for in the influenza virus genome, which is the non-structural protein 1, which we have said absolutely nothing about up till now. So, non-structural protein 1, it actually has a correct name, unlike non-structural protein 2. It truly is non-structural. It is not in the actual virus particles themselves, so it isn't structural. However, when the virus has infected the host cell, it will produce non-structural protein 1. And what's the function of non-structural protein 1? Well, it is to inhibit, and this line means inhibit, the host cell's antiviral mechanisms. So when a cell gets infected by a virus, it doesn't like it, and it can recognize that it has been infected by a virus. You can imagine, this is a major, major disturbance for the cell. It's got all of these bizarre proteins that it doesn't have coded for in its genome, now being produced at huge, great, ridiculous rates. It can tell that something is up here. We don't completely understand the actual proteins involved in doing this, but it does seem logical that this cell is able to recognize through some sort of signaling pathways that something is majorly wrong here, and it does. So the host cell does realize that something is majorly wrong and initiates what we call the antiviral mechanisms, and there are loads of these. However, I'm just going to go through one of them, one of the major ones. So one of the major antiviral mechanisms that cells have is that when they think they are infected by a virus, they can produce these special molecules called type 1 interferons. And there are two different types of type 1 interferons, interferon alpha and interferon beta molecules. So, type 1 interferons. Now, the type 1 interferons are not as famous among medical students and biology students as another interferon. The famous interferon is interferon gamma, which is the one that T helper 1 cells can release, which gives macrophages the power to be utterly ruthless with trying to destroy pathogens. And it's the one that, for instance, is hugely important in the immune response to TB, where the macrophages hugely struggle to kill the TB bacterium, and then they need the T helper 1 cells to come along and throw interferon gamma at them, which then gives them the permission to be utterly vicious with their mechanisms to try and kill the TB bacterium. So interferon gamma is the really famous one, but there is an interferon alpha and there is an interferon beta, and these for short are abbreviated down to IFN alpha and IFN beta, and these are in the group called type 1 interferons. Interferon gamma is not a type 1 interferon. It does very different things to interferon alpha and interferon beta. So as I say, these aren't as famous as interferon gamma, and they're an antiviral mechanism. So when a host cell thinks that it is virally infected through mechanisms that we don't quite understand, it starts producing these type 1 interferons. It starts secreting interferon alpha and interferon beta. So let's just draw a little picture here. So let's say here is our infected cell. So I'll draw it in red because it's not a happy cell. It's going to start chucking out these molecules. So it's going to start chucking out interferon alpha and interferon beta. And these are effectively a warning signal to the surrounding cells that they are at huge risk of getting infected by the influenza virus. They also have functions on the cell that actually releases them itself. So here is a surrounding cell, here's another surrounding cell that might not be infected at this point. So what happens is these type 1 interferons, they're going to work on the surrounding cells, so they'll bind to receptors on the surrounding cells, and they're also going to bind to the equivalent receptors on the cell that actually secreted them itself. So it's going to be local signaling as well as autocrine signaling, which is where you release a molecule for it to stimulate your own receptors. So this is a nice piece of terminology here. when we describe signaling as autocrine signaling, it means that a cell has released a signaling molecule, such as these type 1 interferons, for that signaling molecule to then bind to its own receptors and trigger signaling pathways in itself. Okay, so the interferon alpha and interferon beta molecules, they're going to work on surrounding cells as well as the cell that released the type 1 interferons itself. And what these induce is an antiviral state. They make the cell go into what we call an antiviral state. So they cause the antiviral state. And what is this antiviral state? This is where a cell completely blocks all protein synthesis. So antiviral state involves no protein synthesis. So the cell completely shuts down all its ribosomes, all its transcription mechanisms. It stops protein synthesis completely. And the idea then is that these cells that are refusing to allow any protein synthesis to occur inside them are then rubbish targets for the influenza virus. Of course, the cells don't like switching off protein synthesis because they need to be making proteins all the time. However, in order to stop this viral infection, it's a price, that's worth paying. So, the interferon alpha and interferon beta, they bind to receptors on the surface of the cell, and these receptors activate signaling pathways that induce an antiviral state where the cell completely switches off protein synthesis, and then even if the virus comes into that cell, it's not actually going to be able to synthesize any of the proteins. It's not going to be able to exploit the cell. At least this is the idea behind these type 1 interferons. Now, unfortunately, the virus has realized that cells have this mechanism and has produced a protein to block it. And non-structural protein 1 indeed does this. Non-structural protein 1 stops the production of the type 1 interferons. It blocks whatever the signaling pathways are inside the infected cell that produce the type 1 interferons. Oh, and by the way, it's important for me to stress that when the type 1 interferons acted on the infected cell, they'd also induce the antiviral state, i.e. they'd shut down protein synthesis there, and potentially then save this cell, even though it's already infected by the virus. If it shuts down protein synthesis, then it's not going to produce any more viruses. Okay, but as I say, the influenza virus has a way around this. It has this non-structural protein 1 which inhibits the infected cell from producing type 1 interferon. So in the infected cell, it will produce this non-structural protein 1. It blocks this mechanism by which type 1 interferons are activated to be produced. Therefore, they're not produced, and therefore the cell and the surrounding cells do not go into this antiviral state, which means that the influenza virus can still operate. It can still infect these cells. So that's one of the things non-structural protein 1 does. I just wanted to actually talk about one other thing that non-structural protein 1 does. I said I was only going to talk about one thing, but I'm going to actually talk about two things. One more thing I want to talk about. Now, we will discuss this in much more detail later on, which is why initially I thought I wasn't going to mention this now, but I do want to mention this now, and we'll discuss it in more detail later on. The other thing that non-structural protein 2 does is it downregulates a really important and really famous molecule on the surface of cells, which is the MHC class 1 molecule, also fully called the major histocompatibility complex class 1. So I will write this out in full here. So this stands for major, and then the H is for histocompatibility, massive great word, histocompatibility, and then the C is for complex, and then it's the major histocompatibility complex class 1. So, as I say, when we discuss the immune response to influenza, we will discuss this in much more detail. But at the moment, no. Unless, of course, you know your immunology. If you know your immunology very well, you'll know that this molecule is extremely important in immunology. If your immunology isn't quite as good as it might be, I will explain later on exactly what this molecule's role in the adaptive immune response is. However, no, at this point of the video, this molecule, major histocompatibility complex class 1, it's a molecule that all nucleated cells in our body have. So I'll just put this here. All cells have this, and they put it on their surface. So I'll draw a little picture here. So Oh, I've missed a word out there. It should be all nucleated cells. So the non-nucleated cells, the red blood cells, don't have MHC class 1 on their surface. But most cells in the body are nucleated, and therefore most cells of the body do have major histocompatibility complex class 1 molecules on the surface. So here is a picture of a cell. Here's its nucleus. And now on the surface, it's going to have this protein complex called major histocompatibility complex class 1, and it has multiple of them. It doesn't just have one of these molecules, it's covered in these molecules, and these molecules are present on all cells, all nucleated cells in the body, and they are really, really important for telling the adaptive immune system that a cell is infected by a virus. They are really, really important for that. So their function is in communicating to cells of this powerful adaptive immune system whether or not a cell is infected by a virus. And if they say to the adaptive immune system, yes, it is infected by a virus, the adaptive immune system will kill that cell to spare the rest of the body. So, this is a major antiviral protein here, major antiviral protein, and therefore one of the things that influenza virus likes to do with its non-structural protein 1 is make this protein's level go down in the infected cell, so it stops the infected cell from producing MHC class 1, and therefore aims to stop the adaptive immune response from being able to kill the infected cell. Now, as we'll see when we discuss the innate immune response, humans have outwitted this. Our bodies are winning, and we know this. Influenza virus overall loses to us. In most people, you get better from flu, you beat the virus, so we are better than the virus. So actually, we have a mechanism that counteracts this mechanism that the virus uses to counteract a mechanism that we already had. So the virus says, let's get rid of this mechanism that the human has got involving the adaptive immune response by lowering the levels of MHC on the surface of the infected cell using this non-structural protein 1. We will see that we have special cells called natural killer cells that are part of the innate immune response that then go around the body looking for cells that have low levels of MHC class 1, and they say, if I find a cell with low levels of MHC class 1, that means that that cell is virally infected, and by some clever virus that has lowered the levels of MHC class 1, to avoid the adaptive immune response, and those natural killer cells, they kill the virally infected cell anyway. So we've got a mechanism to counteract this mechanism that the flu virus has, and we'll go into that in more detail when we discuss the innate immune response. So, for now, know that the non-structural protein 1, it's involved in inhibiting antiviral mechanisms that human cells have, and I've given you 2 examples of this. There are more examples that you could go and read up on if you like. One of them is that it stops the production of type 1 interferons, which are a major antiviral mechanism. They're these signaling molecules that an infected cell can release to act on other cells around it and also itself to induce the antiviral state where they shut down protein synthesis to stop the virus being able to use them to make more viruses. Another way that the non-structural 1 protein works is it downregulates the MHC class 1 on the surface to try and evade the adaptive immune response, but as I say, later on we'll see that we have a way of getting around this. We have these natural killer cells that go around looking for cells that have low MHC class 1, and if they find them, they say you're virally infected by a clever virus, but we know exactly what game it's playing, we'll kill you anyway. To spare the whole body, of course. Okay, so there we complete now the viral replication cycle. We'll have a break there. In the next video, we will continue on our discussion of the pathogenesis. We'll talk about how the virus spreads down the respiratory tract, and we'll then start talking about the inflammatory response, and we'll see then where all of the symptoms, the local symptoms, and also the systemic symptoms of flu come from once we've discussed the inflammatory response.